Alright, welcome back. In this video, we're going to go over the Proximal Convoluted Tubule, or PCT. This is going to be a really fun video because I enjoy going over the mechanisms of proteins and how ions move in every direction, so it's going to be a fun video. So the Proximal Convoluted Tubule, or PCT, is the second segment of the nephron, the first segment being the glomerulus. The PCT does major absorption of ions, water, and nutrients. This is like the tank of the nephron. It does so much reabsorption. And what I mean by reabsorption is when we reabsorb things, we are taking stuff from the tubule fluid, which would be the fluid that you would urinate out, and instead taking some nutrients and ions and shoving it back into the blood. That's called reabsorption. Our body is reabsorbing the nutrients and ions back into the blood. So that's what I mean by reabsorption. And the cool thing is that the PCT reabsorbs about 60% of all the filtrate that was filtered by the glomerulus. The PCT can also do some filtering of the blood in regards of hydrogen ions and ammonia, but we don't really go into that. That's not important. What we really care about is the reabsorption aspect of the PCT. So here is basically the kindergarten baby version of the PCT. This is just showing you where the ions and especially water and nutrients is going. This doesn't tell you much. This is not what we care about. But just as an overview, this yellow thing here is the glomerulus, which we went over last video, and the green thing is what we're talking about here, the PCT, or Proximal Convoluted Tubule. So this is just baby stuff. The real stuff is here. Now, this looks very complicated, I know, but trust me, it's actually really easy. But what they do is basically break it down into steps. Now, the one thing I need you to know is that this entire mechanism of reabsorption, and as well as the loop of Henle, which will be the next segment, and as well as the DCT and the collecting duct, which is the other parts of the nephron, is all due to sodium chloride. Because sodium chloride generates this osmotic gradient as well as electrical gradient around the nephron. And this is what pushes ions to one side. This is what causes reabsorption to happen. This causes secretion to happen. This causes a bunch of stuff to happen. And it's all because of sodium chloride. Okay, so to start with this, here's what we're going to do. We're going to break this up into 4 segments. The first segment is this section right here. This is the tubular fluid. So anything in this area, in this section right here, will become urine eventually. If it follows the entire nephron pathway, it will eventually become urine. And it's part of the PCT. So this section is the PCT. Now the PCT is actually made up of epithelial cells, or cells. So the PCT is a tube made up of cells. So the outer layer of the actual tube is made up of cells. And the cells are here, these little box things. So this is the epithelium of the PCT. That's the second segment. The third segment is outside the cell, the extracellular fluid. This comes into play in the next step of the loop of Henle. Don't need to really worry about it now. I will mention something in the coming moments, but don't really worry about it. And the destination, the final segment here, is the blood vessel, where we're trying to reabsorb stuff, like nutrients. Let me give you an example why we're going to do this. For example, if we have low blood pressure, we need to get stuff, ions, and nutrients back into the blood vessels to increase the blood pressure. This is why we do reabsorption. Say if our blood glucose is low. Well, what we're going to do is we're going to steal some glucose from the fluid that would be urinated. Let's steal some and transport it back into the blood vessel. That's the goal, to get our blood sugar up. This is why we do reabsorption. Okay, let's start. We're going to start from right to left. It's the easiest thing to do here. So here's the first thing. This is called the SGLT symport protein, or sodium glucose transport protein. Here's what we're going to do. We're going to take sodium and glucose and transport it together from the tubular fluid. This is the fluid that would be urinated out, and we're going to shove it in to the epithelium cell. This is called the SGLT. The reason is because glucose does not willingly want to go into the cell, but sodium does. So what happens is glucose piggybacks off of sodium's gradient and powers this pump to shove both through at the same time. So sodium goes in and glucose goes in at the same time. They're going in the same direction. This is why it's called a symport. Symport meaning two things going in the same direction. Both things, sodium and glucose, is going in the same direction into the cell, into the epithelium cell of the PCT. All right. The next thing we're going to do at the same time is the sodium hydrogen ion antiport. Here's what we're going to do. We're going to shove some sodium inside, but in exchange, we're going to take some hydrogen ions and pump it into the tubular fluid to be urinated out. This is a mechanism for acid-base balance. Say if our blood is too acidic, what we can do is we can start shoving in hydrogen ions back into the tubular fluid. So the hydrogen ions are in the cell and you need to put it into the urine. What will become urine. So this is a way of acid-base metabolism, or regulation rather. So it's called the antiport because sodium goes inside the epithelium cell and hydrogen ion, which is known as acidic molecule, will go into the tubular fluid to be urinated out. At the same time, we have now the chloride anion antiport. Here is the trick. Wherever sodium goes, chloride will follow. They're best friends. They love to be next to each other because of sodium chloride, of their chemical properties. So wherever sodium goes, chloride will follow. So here's the thing, is that chloride does not willingly want to go inside the cell. So here's what we're going to do. We're going to use a chloride anion antiport. We're going to shove anions, which are basically molecules with negative charge, shove them outside to the tubular fluid, and in exchange shove chloride molecules inside. So sodium and chloride can be together. At the same time, we also got something called aquaporins. Aquaporins are specialized water channels. Only water can cross through there. So water is going to leave the tubular fluid, the fluid that would be used for urination, and it's going to enter the cell. At the same time, so this, I promise, this is the last part here. There's something called tight junctions. You see cells, we have so many millions and millions of cells, but they're not glued together. However, we have some special proteins that actually kind of hold them together so they don't just wander off, but they're loosely attached. So it's kind of like a bridge. Now, it's called a tight junction, this little gap here. And it holds the cells together so they don't wander off, but it's not completely impenetrable. There's basically a hole or a center canal that very small molecules can go through. So water, urea, uric acid, and a bunch of ions can actually go through these tight junctions here. Oops. And they basically can go through this little central canal through the tight junction and end up here in the extracellular fluid. Okay, so let's take account of what we have now. So that's the end of phase 1, like I like to call it. So here we have sodium. Now we have glucose in here. We have another sodium molecule here. We have a hydrogen ion outside. We have a chloride ion inside. And ions outside here in the fluid. and water. Okay. Now comes phase 2. Here's what's going to happen. Glucose is going to use its own specialized transporter protein. It's basically going to go outside and go into the blood vessel just by itself. Nothing special. It just is able to do it now because there's basically a low concentration inside the blood. So glucose will want to go in. All right, so now let me erase the glucose here and put the glucose in the blood now. Ta-da! Okay, now we have the sodium-potassium pump. And here's what's going to happen. The sodium-potassium pump is going to pump 3 sodiums outside for 2 potassiums inside. So this is the importance here. The sodium, some of it is going to enter the blood if we do have low blood pressure. But what most of it's going to do, it's going to stay in this extracellular fluid, this area right here. It's not going to enter the blood vessel. Most of it's going to stay in this blue area. The reason is because it's going to create the gradient that we need for the loop of Henle in the next step. So we got some sodiums here and maybe it's like sodium here. Let's just say 2 sodiums there. It's not, I shouldn't say that actually, but it's just some number of sodiums. At the same time, so potassium is going to be pumped in. So we got 2 potassiums being pumped in here. Okay. Now here's the thing. Chloride needs to be with sodium. It really wants to be with sodium. Because remember, sodium chloride. But chloride cannot willingly leave the cell. It doesn't want to do that. So what are we going to do? Well, there's actually a high concentration of potassium inside. And there's a low concentration of potassium in the extracellular fluid here. So by process of diffusion, potassium will want to go outside into the extracellular fluid because there's a low concentration of it. So it's going to use this gradient, this electrostatic force, and shove chloride outside. And this is called the potassium chloride symport. So potassium is going to use its power of diffusion to go outside and join this fluid here. Let's just do 1. And chloride is going to use that power, the electromotive power here, and actually go outside the cell. So sodium and chloride can be together now. They're happy. So now we've got rid of the sodiums and we got rid of the chlorides. Okay. This figure is a little wrong because there are actually aquaporins as well. But the aquaporins are only in special locations that we don't really care about here. But it's going to become important in the loop of Henle. So don't really worry about this water here. Just know that there's an aquaporin here, and water can enter the cell. The water can actually leave in the loop of Henle, and we'll talk about that. And that basically concludes the PCT. Here I have some extra notes here, basically a summary of what happened, the first stage and the second stage. So first, the sodium glucose symport, SGLT protein, takes both sodium and glucose from the PCT fluid, or tubular fluid, and shoves it into the tubule cell. At the same time, the sodium hydrogen ion antiport is pumping sodium into the tubule cell and a proton in the tubular fluid. At the same time, the chloride anion antiport is pumping chloride into the cell and anions into the tubular fluid. And water enters through aquaporins in tight junctions, as well as some ions. Stage 2, sodium-potassium pump is pumping 3 sodiums into the blood and 2 potassiums inside the cell in exchange for ATP. The potassium chloride symport is pumping both ions from the cell to the blood. And glucose exits through its own channel into the blood. So this is basically a summary of this. So if you understood this, don't worry about the summary. But yes, if you found this video helpful, please like and subscribe. Until next time, later.